Welcome to the Spark Live webinar series, one component of our Spark Knowledge Mobilization Program. Spark is Children's Healthcare Canada's shared platform for advocacy, research, and knowledge. Spark Live is where we gather each week to curate, convene, and showcase excellence and innovation from across the child and youth healthcare community. Our goal is to spark conversation, ideas, and action. Children's Healthcare Canada would like to thank our funding partners for their ongoing contributions that support all of our programs and activities, including this Spark Live bi-weekly webinar series. There are two options to join in on the live conversation. Questions and comments for our panel or presenters can be typed into the question box, or comments that you want to share with the audience can be typed into the chat box visible to all of our attendees. For those of you on Twitter, tag @ChildHealthCan on any webinar-related tweets or use the hashtag SparkLive. And to keep up to date on all of Children's Healthcare Canada's webinars and other activities, be sure to sign up for our weekly Spark News e-bulletin by visiting our website at childhealthcan.ca.
Hello everyone, and welcome to another edition of Children's Healthcare Canada's Spark Live webinar series. This month, the Spark theme relates to child health research. I'm Paula Robeson, your host for the next hour. Children's Healthcare Canada acknowledges that our offices, located in Ottawa, are on the unceded, unsurrendered territory of the Algonquin and Anishinaabe people. We recognize and deeply appreciate their historic connection to this place. We also recognize the contributions that Métis, Inuit, and other Indigenous peoples have made in shaping and strengthening this community, our province, and the country. Today, during Child Health Research Month, we're delighted to bring you this webinar, Can We Protect Canadian Children from Unsafe Drugs? Before we begin, I invite you to answer a few polls that Jose will now pop up for us. Please note that you'll have to answer all three of the poll questions in order to submit your response. And while you're entering your responses, I'd love to introduce our speaker today, Dr. Bruce Carleton. Dr. Carleton is a professor of pediatrics, medical genetics, population and public health, and pharmaceutical sciences at the University of British Columbia. He's a senior clinical scientist at the BC Children's Hospital Research Institute and director of the BC PHSA Therapeutic Evaluation Unit. He was a member of the Canadian Agency for Drugs and Technology in Health Drug Expert Advisory Committee and has served as an advisor to Health Canada and the US FDA. Dr. Carleton's research focuses on improving the safety and effectiveness of drug therapy in children. Just a reminder that we record all of our webinars. As mentioned in the introductory video, please type your questions into the question box at any time. I'll prompt you via the chat as well, and I'll pose your questions to our guests following the presentation, unless there are clarifying questions that we need to interrupt his presentation for. It's now my pleasure to pass the mic to Dr. Carleton.
Welcome, everyone. Thanks for joining, and I hope you enjoy the next 30 minutes or so of time with me and a little bit about the work that we're doing here. So you've heard the topic. Dr. Michael Reeder is not yet available, but I hope to see him soon today as well to participate in today's webinar. This is really the case with drug therapy. I think that it is probably a misnomer to call drugs safe or unsafe. Instead, I think we should say that there are right drugs and wrong drugs for individual patients. These types of reactions can occur in children and adults and are often drug-induced and can be fatal. And so the question is, why is a drug that is widely used and helpful for many without tragic adverse events like this, which often lead to death with mortality rates of approaching 70%, when the vast majority of children or adults that take them wouldn't have this reaction? So what makes those particular patients different and subject to these tragic reactions? That's really the nature of the work we've been doing. And adverse reactions are a major problem. Drugs, again, are one of the great gifts of humanity to humanity, and something that we've worked very hard to to help develop for the benefit of society, but they also have tragic consequences. And you can see all of the costs involved in managing adverse effects, or side effects, as they're often called, I think inappropriately. I think we should call them what they are. And most of the tragic, serious reactions aren't known until the drug is on the market, because they are quite actually infrequent. But frequency and rarity mean nothing if the previous picture was your child, and you're faced with the grim reality of the situation. Most adverse reactions are never reported, and so we do have some new legislation in Canada, the Protecting Canadians from Unsafe Drugs Act, which has increased reporting. The question is, what do we do with that information, and how does it actually help to make drugs safer in the future? The paradox of drug development is that we study the population at the top, and we actually treat the population at the bottom. And we do this specifically not because people are studying the wrong populations, but we want to understand the biologic efficacy of drugs, how they work, and in general what their safety is in a defined population. And the more narrow we define that population, the better we can understand the biology of those outcomes. But in reality, we treat patients that have many comorbidities that we didn't study, have different levels of renal function, are at different ages, very young, very old, very frail, and those conditions are often not studied in trials. And so we end up with, as Health Canada has stated in 2007, 50% of serious adverse reactions becoming known only after the drug is on the market. So drug development itself isn't really able to capture these outcomes. And so we are faced with a post-market evaluation that we must continue with over time. And the idea of pharmacogenomics, taking pharmaceuticals and genetics and combining the sciences together, is to be able to screen out patients that are at high risk for adverse reactions and should never get the medication. And then patients at low risk would know that it would be quite safe to continue with therapy. And in patients in the middle, we throw up our hands and say, we don't know. There's an increased risk, but it's not perhaps enough to warrant not continuing. But perhaps we'll use a lower dose, since most drug reactions are dose dependent. And that's the concept of pharmacogenomics. And I think it has a lot of promise. And there's actually more than just the predictability of adverse events in patients that really highlight the value of this science. I'm going to get into that in a moment. This is an example of the metabolite thought to be primarily responsible for fulminant hepatotoxicity, liver failure in children who take valproic acid. And these are the metabolite levels in different ages. And so you can see that in pediatric patients, the considered hepatotoxic metabolite is actually quite a bit more prevalent in children than it is in adults. And so this reminds us that children are not small adults. They are different metabolically. They biotransform drugs differently, and they are at a different risk profile than adults, and why we need to study drug use in children and not avoid those studies, because that lack of study can put them at risk. And these data clearly show that the at-risk populations are young children versus adults. About 2004, we began a project then called GATC and now called CPNDS, the Canadian Pharmacogenomics Network for Drug Safety, deciding that we would actually tackle this problem of drug safety and find solutions to these problems, not just catalog adverse responses, and really focus on children, which is needs to be a considered focus point for this type of work. And so we began with a hypothesis that there are genes that control drug function in the pathways of biotransformation in patients. So as they absorb the drug, as it's distributed throughout their system, as it's metabolized and then eliminated, those four phases, absorption, distribution, metabolism, and elimination, are all controlled by genes. And if we understood which genes controlled which area, which biotransformation pathway, we could begin to characterize genetic risk in individual patients and come up with dosing guidance and plans to predict the safety of medication and avoid these serious ADRs altogether. That was our thinking in 2004. And I'm going to show you what we've done since that time. So the idea was fairly simple. You find a child with a serious adverse event. You find a child that got the same drug without the serious adverse event, and you compare the genetic profiles. Now, of course, one child in each of those two groups is not sufficiently powered to derive any generalizable conclusions, and so you need larger numbers. And that's what we began doing, is amassing DNA samples from children across Canada and the world, because no one else was doing this at the time. And so people began sending their samples as well. And we collect DNA, we review the adverse event, we carefully characterize within the CPNDS network what's happening. It's not just a physician's report or a patient's report. We need objective data to understand the nature of the reaction in great detail. Then we screen for genetic variants in those two populations, fairly sure that we have a population of patients that did experience the adverse event. It wasn't due to another cause compared to another drug-exposed patient exposed to that drug who did not have the reaction. And again, those two populations compared with proper statistical power can lead to some pretty interesting conclusions. And then, of course, replicating our findings in even larger populations or other populations to understand further the generalizability across global populations is really important. So we set up a network. I remember very well doing this in 2004 and meeting with a lot of senior medical directors at children's hospitals and administration officials. Administration officials in general were, their comment was, it's about time. It's about time we did this. This is a great idea. Senior medical directors thought it was a great idea, but many felt that we would not be able to find any adverse reactions, or at least they wouldn't be able to help us find any because they didn't see them. And that really was confusing to me, because the literature is filled with examples of large numbers of serious reactions. And yet clinicians that should know about these were unaware that they would be able to help and find the numbers possible to make powered analyses. These are rare events, so it's reasonable to assume that the events would be rare. I was just shocked that they didn't think they could help. Many said that they thought that it was because they were very careful with prescribing, which they probably are, and therefore they didn't see as many as perhaps might exist out in the community. Well, the numbers don't lie. So there are a lot of adverse reactions that are serious in children in Canada. And we've collected more than 10,000, almost 11,000 cases of adverse drug reactions. We have nearly 100,000 controls, which is wonderful. You want a large control population for your comparisons to understand generalizability in a population. So that's worked very well. And it takes quite a bit of time to do this characterization. I said it was careful, and I want to emphasize that these things can take days to characterize appropriately. There are lots of variables to consider. And so this is not just simply 5 minutes of communication. The reality is that we are 99% genetically identical as human beings, but anyone can see these 15-year-old boys don't look anything alike, even though they're the same age. And that 0.1% difference between us defines all of the characteristics that make us different. And in fact, the question became, well, what area of the genome would you look at to understand variability in drug response? And the answer is the SNP, or the single nucleotide polymorphism. Most of the variation in DNA, about 90%, is in these polymorphic variants throughout the genome. These are letter changes in the amino acid sequence. There are 4 letters in the human genetic alphabet: G, A, T, and C. That was the first name of our network, GATC. And when those transformations occur, you have a gene variant. And those gene variants can have very different function. They can have reduced function. They could have enhanced function. They could or they could have no function, and those things can code for drug response. And that's what we began looking at. And we can actually look at large numbers of these with computers and sequencing technologies today. Hello, Dr. Reeder. Nice to have you with us.
Hello, Bruce.
Let's continue along here, and please add whatever you'd like as we go. So now that we know that we're looking at SNPs to look at drug response variability in children, we have to decide what SNPs to look for. So the first place is to go to the literature, and in 2004 there wasn't a lot, but we began looking to see what SNPs, what gene variants had been defined by others. And we looked specifically within those 4 phases of drug absorption, distribution, metabolism, and excretion. We found about 2,000 that we felt were good targets broadly for drugs across the spectrum. Right now, our variants are around 10,000, so we're looking quite a bit larger now. And there are sequencing arrays that are 2.5 million, 5 million, et cetera. We often do 7 and 8 million genetic variants through imputation examination of that many variants. The problem with more, though, is that you're doing multiple testing, so you just find more significance just by chance because you have so many comparisons to make, and so you have to correct for that. And so that's one of the challenges of that. So it's really important to start with a focused approach of the specific drugs, candidate genes, and then to move forward from there. So I want to next talk about a case, and this is a 10-year-old who was previously healthy, presented at our children's hospital with a neuroblastoma that's biopsy confirmed, and began the standard protocol for the treatment of his neuroblastoma with doxorubicin, which is an anthracycline known to cause heart failure. And the dose is cumulatively limited to 300 milligrams per meter squared to prevent the probability of heart failure. And because it is quite significant, and dose is the most significant predictor of failure. And yet, even with that, we still have kids that have heart failure. So it's not the only predictor. So we felt like we could probably find relevant things here in these patients that could help us better understand who is at risk. And so at the end of the last cycle of treatment, this child was undergoing a routine CT scan to check on the status of the tumor, developed serious cardiac function, in fact, had no cardiac output, was intubated, rushed to the ICU, placed on ECMO. The heart was essentially non-functional. The child received a heart transplant. That transplant, unfortunately, was rejected, a terrible outcome, and received a second transplant. So this child's cancer was treated, but was left with this life-shortening outcome of cardiac toxicity. So these drugs, again, are really important in the therapeutic treatment of tumors in children, and are used in about 70% of all children with pediatric cancer, and between 50 and 90% of women who get breast cancer as adjuvant chemotherapy, add-on chemo. And so about 1 million patients receive these drugs every year. They are highly effective agents. But despite that, we've known since 1967 about this. And I wonder sometimes where we are when we can say that we've known about the toxicity since 1967 and we're still managing it. We're not actually solving the problem, we're just still dealing with it. So this is a particular problem, and probably more of an issue in children because they have smaller hearts, smaller organs, they are smaller biological organisms, and the mortality rates are quite high. These data come from adult patients, but they tell an interesting story. So the dose of anthracycline exposure is on the horizontal axis, and the probability of heart failure, cardiotoxicity is on the y-axis. And you can see that the probability exceeds 20% at around 650 milligrams per meter squared, or 600 milligrams per meter squared. That's the cumulative limited dose in adults, 600. And so in kids, we give them half that. It's a fairly blunt way of defining toxicity. But I think the important point here is what I've yellowed on the graph, which is that there are patients that get much lower doses than that, and in fact are still get heart failure, even though they don't reach the cumulative the dose limits that generally relate to heart failure. So with our CPNDS network across Canada, we took 266 children who had absolutely no evidence of cardiac dysfunction, despite full treatment with anthracyclines, and had received at least 5 years of follow-up. So we knew that they didn't even have any late toxicity from this drug after 5 years and within 5 years. And then we took 78 patients who had very significant cardiotoxicity during or following treatment, and we compared the genomes of those patients. And what we found were some really interesting findings. The first is SLC28A3. Any odds ratio or OR that is less than 1 in this particular instance means it's protective against cardiotoxicity. So we found a protective variant, something that protects patients and something that we hope to exploit in the future to the benefit of patients who are at higher risk. So this was significant. It was discovered with a Vancouver population, replicated with populations of patients across Canada, and a second replication done with colleagues in then Amsterdam and now Utrecht, where the patients in the Netherlands are treated. The mechanism here, this is a cardiomyocyte. The mechanism is this is a transporter. And when you have reduced expression of this particular transporter, you have less of these drugs into the cardiomyocyte. You develop less reactive oxygen species and toxic alcohol metabolites, and therefore have less toxicity. And so the idea here is that we don't need the anti-cancer drug in the cardiac cell. We need it in the tumor cells. And so if we can prevent it from entering cardiac cells, we can prevent the toxicity. That's the potential here. We also found a variant in UGT1A6 that put patients at between 4 and 8 times higher risk of cardiotoxicity. And this was quite interesting. Again, in these same populations, we know that this drug is a glucurononyl sulfyltransferase. It actually glucuronidates many different substrates, meaning that it is involved in the metabolism. But anthracyclines aren't metabolized. It was Dr. Reeder's work in the lab that helped us to understand that actually it's not the parent compound that's creating these that are glucuronidated. It's the metabolites. It's the byproducts of biotransformation that are actually glucuronidated. This suggests then that the parent compound, the parent drug that we administer, isn't toxic. It's the metabolites of that compound, and this leads to the accumulation of these metabolites. They're toxic and they create cell death. We then completed a genome-wide analysis and found another variant that puts patients at about 5 times the risk overall. And this is a variant in a receptor variant. So we have a transporter variant that transports the drug to cardiomyocytes. We have a metabolic variant that creates the metabolic toxic compound. And then we have a receptor variant that interacts with the receptor that also is responsible for a 5-fold increase in toxicity. So looking at these together, we know that we've come up with very significant findings about why these patients who have toxicity biologically experience that. And we began a series of steps to understand the role of these variants in these particular cells. First, looking at human cells derived from these patients, where we would take peripheral blood and we would grow cardiomyocytes and expose them to these different, to this drug class in the lab, in a cell model, to look at cell death and to understand that we have not just an association, between the gene and the drug outcome, but that actually the gene variant itself is responsible for the toxicity. And this was shown in a paper published in Cell Stem Cell some time ago. We then began looking at managing heart failure in these patients using all-trans retinoic acid, which is really a fancy word for vitamin A, by the way, but using vitamin A to protect cardiac cells in these patients. And we were actually successful in doing that. These data show that that in the doxorubicin treated patients, their cardiac function, which is the vertical axis, is the lowest. The green line shows their cardiac function is affected by the drug. When we give them vitamin A, these mice, we can actually preserve cardiac function more like patients who've not been exposed, closer to that level. So this is also significant. And then third, we wanted to look at this protective variant, and this has been published in Circulation just this year. We wanted to look at this particular protective variant, and can we actually increase cell viability when patients are exposed to this drug, and by using another drug that can actually enhance this particular transporter that is protective, and we can do that. And so we have a marketed drug that has, in which we have human data that we can actually protect the heart. So now we're beginning to move into that phase from looking at adverse events, finding the genetic constituents, and now finding therapeutic intervention strategies to prevent harm. And so this modeling that we're doing now that puts patients, we can now identify, for instance, with anthracyclines, we get what risk patients are at. The lowest possible risk is 14%, which seems high if you ask me. But again, we're using potent pharmacologic agents at high dose to prolong life in pediatric oncology. So you're not going to see 1 or 2% rates of adverse reactions. You're going to see higher rates than that. And the risk goes up depending upon the number of risk variants that you possess. And so we can now produce these models. We do this in 10 centers across Canada, showing clinicians and patients what their risk is compared to other treated patients, which is actually the right population to compare to, because we're going to treat them. We have to, because they're facing a life-threatening disease. And this is the first step at identifying the patients at higher risk, and therefore the patients that should be part of clinical trials designed to reduce the toxicity of this important agent. So we have 10 pediatric oncology centers across the country in which we're delivering these results, and we're using this information to further refine our understanding within the network of how best to manage adverse reactions. And I thought now would be a good chance to just show you a quick video about one particular patient and the story of this particular patient using a different drug. I often say that at the Children's Hospital, we manage life. We don't manage the end of life, we manage the beginning of life. Adverse drug reactions, or side effects, are the 5th leading cause of death in North America. And what makes one patient experience a tragic side effect, and yet another patient who receives the same drug at the same dose has no adverse effect? Now we need to sort of incorporate human genetic variation so that we can understand the diversity of how cancer affects children and then come up with better treatment options. Children, for the most part, are an unstudied population of patients when it comes to drug therapy. They only are 25% of the population, and yet they are 100% of our future.
Rory was born with a teratoma, and that's a type of a germ cell tumor. It's a solid tumor. She had surgeries and procedures and some complications. You're kind of thinking, how is she going to be able, the little body, to handle all these things and all the possible scary side effects, not only if she survives these treatments, but if the long-term side effects when she goes home and if she gets to grow up, what that's going to look like. I wanted to give her the best outcomes if we could. If we could know more information, we could plan ahead and hope to support her better. I would have never thought to even ask these questions before the genetic study.
Genomically speaking, the risk of hearing loss is between 0 and 88%. And in Rory's case, it was that high-risk grouping that caused us to stop and consider alternatives. And in her particular case, use an alternative drug which allowed her to preserve her hearing and yet still receive effective anti-cancer therapy.
And for parents, information is powerful. That's what they need. That's what we do as oncologists every day, is to give families information. This study, it's changing how we approach medicine, and it really is taking in family values a lot more. And by giving them all this information, they really are a partner at the table with us as oncologists. This project is actually being now launched across Canada.
And if it's good enough for Canada's children, is it not good enough for the world's children? And that's the global reach of what this project is all about.
Now she's full of energy and able to get out and live life to the fullest. She's retained all her hearing, and she is the most curious, feisty girl now to this day. You can't hold her down. Every day that she can be out exploring and just loving life is a big gift.
Sorry. So I hope that gives you an understanding of the types of work that we're trying to do at the individual patient level, as well as across all patients, as the network progresses into its 20 years of activity. And the problem with these networks, and this is a challenge to all of you on the phone that are part of Children's Healthcare Canada, is how do we fund these things? I've raised from federal and provincial sources, close to $90 million since 2004, but this isn't an endless tap. These granting agencies like this work. They've funded it willingly and supported it greatly, but the tap is at an end. So how do we continue this type of work? And what responsibility do we have as institutions to do that? Well, we do have a network. We do characterize adverse events. There is now a mandatory requirement for reporting serious adverse reactions and medical device incidences by hospital, and we can help meet that standard. And so, I've yet to hear from any institution saying, "Oh, we have this mandatory requirement now from Vanessa's law, and how can you help us meet that?" But we certainly can do that. In fact, we collect the data. So that's something that the network can help you all in various pediatric institutions meet, and we'd be happy to do that. But reporting is one thing. Finding solutions to drug safety problems is more complex, and it requires more effort. And I don't want to stop at just reporting and thinking that others are going to solve this problem. I think I've adequately described the complexity of the task ahead, and yet the opportunities as well. And I think that instead of just predicting who's at risk, we've really moved into a phase within the network of focusing on the mechanistic basis of adverse reactions, why these reactions occur, and develop real solutions as a result. And they're coming. Vanessa's law requires that we collect post-market safety data. That's what CPNDS does every day. It suggests that appropriate action will be taken when a serious health threat is identified. And beyond warning people of an effect, I think CPNDS moves us into action beyond that by looking at interventional strategies to actually prevent the toxicities. That's really where we need to move, not just warn people, but find a solution. And of course, Vanessa's law also is aimed at promoting greater confidence in the oversight of therapeutic products by increasing transparency. It would be great if we could have participation at the funding level from Health Canada for a network like this. That would make me feel better, and I don't have to be the head of that. I'm just interested in promoting the safe use of therapeutic products by finding solutions to these problems. Again, it's not safe drugs or unsafe drugs. It's right drugs and wrong drugs for individual patients. So lastly, this is the serious ADR cycle of Vanessa's law, where care teams and patients document a serious ADR. That's what CPNDS does every day. We're supposed to then report them to Health Canada, and then Health Canada communicates safety findings to hospitals, and then we share again the learning. But my issue is, do we want to go beyond just saying this drug can cause this reaction or these patients are susceptible, and find solutions? That's really what CPND is all about. Thank you very much for your attention today. I'm happy to take questions.
Thank you, Dr. Carlton. Dr. Ryder, you noted in the comments that you had a few slides you wanted to share as well.
Just a few, just to help put perspective on things. I can't share screens right now because Bruce is still sharing. So
I will take that away and you can. There you go.
Thank you, Bruce. Great. So gotta click this back up. So just to perspective, I think first of all, Bruce has given a brilliant talk on the work that's been done on CPNDS. And just to put perspective on things, just a conflict of interest slide. I think as child health caregivers, we have to think about perspective. One of the perspectives is, let's look at where we came from. So in the 1930s, this is data from the CDC and Health Canada. About 1 in 5 children died before they were 5, and there was almost all due to infection. And now it's 0.5%, like 1 in 1,000 children die primarily due to the complications the first year of life. 3 things change it: vaccinations, sanitation, and drug therapy. But it's a double-edged sword, a sword with 2 blades, a good part and a bad part. And unfortunately, this is actually the 42nd year after Lazaro's epic paper describing the burden of adverse drug reactions published by Lazaro Oncology at U of T, in which he estimated that 100 to 150,000 patients a year in North America die of adverse drug reactions. In the 42 years since then, I would have to say the progress has been disappointing in moving things forward. Further to Bruce's comments, if we've known this problem for 42 years, why haven't we done more about it? Because inside every course of therapy, there's an ADR struggling to get out. And you would think because of the way that drugs are regulated, this would be taken care of in the regulatory process, but it's not, because the regulatory process isn't linear and tidy. This is David Lee's lab in Health Canada. It's circular and complex, especially for pediatrics, because all kinds of drugs come on the market for adult use, get used in children, and never get properly evaluated. And as Bruce noted, adverse drug reactions are actually a problem, because in pediatrics, they're probably the 3rd or 4th commonest cause of death. Certainly in the general population, it's about the 4th commonest, but that's never reported because of nosology. And I also have an example here to share. So a child gets nephroblastoma, gets chemotherapy with ifosfamide. Our lab has shown quite convincingly that one of the side effects of that is actually renal failure related to how the drug is metabolized. And knowing that you probably don't share my love of organic chemistry, I'm not going to get into side chain versus core oxidation, but basically some children get renal failure. So over the next 10 years, the child grows, the kidney doesn't, and the tumor, despite the fact there's no tumor, there's also no growth in the kidney. Child goes on dialysis, and dialysis is a dangerous place to be because the 5-year mortality on dialysis is about 80%, mostly cardiac, and the child dies of complications. The cause of death is nephroblastoma. So why is that? The bizarre fact, because there's not a molecule of nephroblastoma left in this child. The oncologist didn't want this outcome, but this is the way these things are coded. So we underestimate the burden of adverse drug reactions, especially serious ones, all the time. Culturally, people came to accept it as a cost of doing business. So Bill Evans and Mary Relling, 2 colleagues and great researchers at St. Jude's, would say that, no, we can prevent ADRs. But a lot of oncologists feel it's the cost of doing business. I think pioneers like Dr. Rakesh at UBC and other people, working with Dr. Carlton, have recognized, no, this shouldn't be the cost of business. We can do better. We have known risk factors that are present in children, and the rate of ADRs in children, despite the myth, actually is probably the same as adults or higher. And we've been using passive surveillance, which has not served our interests well. We talked about Pimlin already, but we would have got Pimlin off the market 25 years before we did if we'd actually looked for this. So we talk about drug allergy, which is really overworked use. It's kind of like saying I have a red car. Okay, those are 2 red cars, but they're obviously very different red cars. So we kind of use terminology sloppily and we don't pay proper attention. And my concern, of course, one of the concerns is where we're going in the future. So in the future, we're going to as we can be following therapeutics now, we're getting much more directed much more molecular targeted. So things like the most expensive drug in the world, which is Almexica, $2.5 million a dose, are coming out in CAR T-cell therapy, all of which are expensive. And CAR T-cell therapy has an adverse reaction rate that is close to 80% for serious adverse drug reactions. So how will we deal with this in the future? I think the future is the undiscovered country, but I think we need to think together as child health providers in Canada. We have a unique opportunity in Canada, I think, because as you know, Canadians institutions can work together in ways that people don't seem to in the rest of the world. I think we have an opportunity to network and get together. Actually, further to Bruce's point, if it's good enough for Canada's children, should it be good enough for the world? It's my contact information. And at this point, I'd like to bring us all back to why we're all here, which is the care of Canada's children. And with that, I think we should open it up for question and discussion. I'm looking forward to hearing what people have to say.
We do have a few questions. Hal asks, are peds oncology patients who receive anthracyclines, that's probably a mispronunciation.
Oh, you got it.
Okay, screened for the genetic variants you describe, and if not, why not?
Yeah, so they are at BC Children's Hospital, every one of them. They're not at every center across the country. And the reason for that are that this is new. And, you know, we're humans. We don't like always to do new things right away. We divide ourselves into groups of innovators, early adopters, you know, early majority, late majority, laggards. Laggards are the people that only buy touch tone phones because you can't buy a rotary dial phone anymore. And so it's a process of getting people to understand this. And to Lynn's questions that she's asking appropriately in the chat, you know, this is an issue that that I find quite fascinating. There are clinicians who feel that letting parents and patients know about their risk complicates the treatment, because patients and parents start asking questions about the need for therapy. If it's going to cause toxicity, you know, what are the alternatives? And there aren't any alternatives, so why inform them? That's, in my opinion, the wrong way to respond to that. I think that what we don't know can hurt us, and we should have open discussions about this. And life is about risks, and life is about trade-offs, and so, you know, as we move this work forward and publish in high-quality journals and produce guidelines and continue to build the evidence base, it will be harder and harder and harder for people to avoid the consideration of pharmacogenomic findings.
I think Bruce has raised important issues. I mean, I've always been a big fan of empowering patients, and actually for the past 10 years, whenever I see a patient consultation, I actually send a copy of my consult letter to the patient, figuring that the family is probably the best custodian of the medical knowledge. You know, I think has been true that people have been reluctant to do this. I think that attitude hopefully will change. But the bottom line is an empowered patient is a better patient. And it's actually important to have the discussion in an open and transparent way, because there's a lot of misinformation out there. And the only way to combat misinformation is good information. The other reason people don't do it, to be blunt, is funding. I mean, because you know, most pediatric hospitals operate with the Globe or a certain fixed budget. And within that fixed budget, if you do test A, you don't do test B. And convincing, in Canada, we've had a problem in, you know, that we don't really, as opposed to like the US, which actually has a separate branch at the FDA dealing with pediatric issues, we don't have that so much in Canada. And it's been a little more difficult to advocate for pediatric causes. So for instance, thiopurine methyltransferase genotyping, which is established by Dick Wenschenbaum, established that when I was a graduate student, you know, so long ago, they were still cooked our food or open fires. I mean, that was established like 40 years ago. That has only in the last kind of a decade come into routine clinical, being sort of supported by the payers, either provincial governments across the country in terms of paying for the testing. So it takes a long time to convince skeptical governments to adopt new tests. And I think that's why networks like this are important, to get robust data, because one thing that in pediatrics, we have the challenge of small numbers, but networking addresses small numbers. But, you know, it's a multifaceted question, a good one, but I think it's a combination of how we structure and how we do things.
Well, I think we have to stop making that excuse personally. I hear it all the time. We don't have funding. We don't have funding.
Yeah.
It's an easy answer to the question. So let's get the damn funding.
Right.
So the issue of funding is that these tests are much more expensive when they're done in small numbers than they are when they're done routinely and when we have scaled technology to do this. But when we're talking about a population of humans that's 25% of the country, and we talk about a rare disease like pediatric cancer, which is rare by even when you consider all tumor types together, you're not going to get commercial labs interested in testing tiny numbers of patients and investing in all the technology that's required. So we've got to make a commitment to this. We have to move forward. The science is there, the data are there, and it's time to make those commitments and decide what the thresholds are. And if you want more evidence, then tell me what evidence you want. Don't just say you want larger trials. Tell me exactly what you want, and we'll create that, and then we'll do that. But the commitment to funding has got to, and the commitment to that as an answer has got to stop. That's what we have to do. And to Lynn, who's a caregiver, and her comments, we don't take adverse events seriously. We even call them side effects. Oh, it's a side effect. Well, it's an adverse event. It's drug-induced harm. Let's solve this problem. That's what we need to do. And because patients, increasingly our health system is supposed to be patient focused, maybe that's the solution to this, is we need our parents and our families to advocate for this in a very strong way, because politicians listen and bureaucrats listen to politicians when voters speak. And that might be the way to deal with this.
Dr. Lewis, I know it mentions the issue of funding, which is true. I think one of the funding issues that we have to address is, it's not, all decisions involve risk, including the decision to do nothing. So when I was a grad student, C. Spielberg actually had that sign in his office that everything involves risk, including doing nothing. And part of the risk of doing nothing for pharmacogenomic testing is there's actually a cost for not doing it. Because I think, if you think of the cost, for instance, of the patient with anthracycline-induced cardiotoxicity, if you could prevent that, you save the system a bucket of money. And increasingly we're seeing, and there was actually an interesting editorial I read a couple of months ago talking about how not doing pharmacogenomic testing is actually cost inefficient. You actually cost the system more. And I think getting the data, bringing it forward is actually the way to address this issue, because I think if you look at the cost of one test, you don't look at the system cost, and the system cost for adverse drug reactions is very high.
Thank you both. Before we take our last question, I'm just going to ask Jose to launch a final poll. And while you're answering those questions, and you'll need to answer all 5 to finish the poll, I will pose the very last question, which was from an anonymous poser, wrong word. When a patient is found to be at high risk of developing, in this case, ADR, to the first choice drug, how would we justify using the second option, which is understandable would be less effective, more costly, or less safe?
Yeah, exactly. That's a wonderful question, and why I turfed it to the public forum, and not just to respond, because I think it's really not just respond in writing, because I think it's really an important issue. So we've got first choice drugs, and now all of a sudden we find the patient's at risk. So what do? Well, welcome to the world of therapeutic decision making, and there are trade-offs. There are trade-offs all the time that we have to make in life, and our goal here is not to make the effective drug less available or say that it's a bad drug. Our goal is to say, how do we solve this problem while preserving that benefit? And so I think that's a first step. Are there ways that we can enhance protective genes in patients that don't have them to prevent toxicities, the SLC transporter, for example? And we can do that with, interestingly enough, dizipramine. Dizipramine is a tricyclic antidepressant that has long been not used, partly because of the available of other antidepressants, but also because of its potential to cause long QT syndrome in children in particular. So the solution to this presents another toxicity. Although long QT syndrome is something that's easy to monitor and probably could be managed in a dose-dependent fashion, these are the questions that we have to continue to work on and find. And I think that as we do this, we will discover that there are either new compounds that we can be thinking about altogether, or experimental strategies designed to protect patients. But the risk and reward are on the same paradigm. And when we drive a car in the snow, we have a higher risk of accidents and injury, but we still drive them often. And so the question is how we manage those risks. But it's fundamentally, that's fundamentally the question. How do you justify that? Well, we have to go through the process and make decisions. And there's never 100% certainty in anything, and there are always risks. So,
I think that's an excellent question to unpack, because that's a rich question, because you could spend an hour, actually a day, debating it, because drugs become second choice for a bunch of reasons. Sometimes they don't work as well, and that's a cold, in the cold light of day, you may have to make the decision. Sometimes they're just more costly, and I've seen this, because I've done a fair bit of consulting work on the adult side with my adult colleagues, and oftentimes drugs are second choice because they're so much more costly than the first choice. Well, in an era of high cost drugs, it seems to me that this kind of testing is important, because it says, well, we'll use the first choice, but we're not going to because it's more, because you're at risk, or it's second choice because it's more difficult to administer. So it depends why it's second choice. But I think, given the myriad of reasons to make a drug first versus second choice, I think this kind of testing and data is important, because it helps us to make difficult decisions in times of therapeutic uncertainty. I mean, as Bruce said, therapeutic decision making is making the best decision with imperfect information. And I now, in the area of molecular, genetic, and high cost drug therapy, these are difficult times for the moral caregiver, because we're making hard choices based in part on economics. But I think if we can base it on data, what's the outcome? Because it shouldn't be about access; it should be about what the best data is.
Well, thank you both. That's the end of our time together today, but there's been lots of engagement in the chat, so the conversations may continue after this. Thank you both, Dr. Carlton and Dr. Ryder, for your sharing your time and expertise today. Thank you to all our members and to other attendees for choosing to join us. We know how busy you are right now and really appreciate your time, and we hope you depart with some practical knowledge to bring back to your organization. Join us for our upcoming Spark offerings. On April 6th, Carrie Robinson of the Public Health Agency of Canada will be presenting on FACT's approach to vaccine confidence in COVID and beyond for children. And on April 7th, Children's Healthcare Canada is hosting an invitational pop-up event focused on mental health leadership. It's always great if you can watch live, as your questions and comments really enrich the discussion. But if you can't, the recordings of these sessions are made available after the fact on our Knowledge Exchange Network. So thanks again for joining us today. Thanks to Dr. Bruce Carlton and Dr. Mike Ryder for this presentation. And hopefully we'll see many of you back here next week. Take care. Bye-bye, everyone.